Good afternoon. Can you hear? हॅलो, ऐकू येते? कॅन यू हिअर? जागृती, पूजा, श्रुती, निकिता, कविता, वर्षा, हर्षाली. Can you hear? Okay. So we'll start with the last point of the third unit. Before that, I've already given you the schedule of your test. Your first test will be conducted tomorrow. सो लेक्चरचं टाईम मी तुम्हाला सांगेल. त्याच्यानंतर 2:30 वाजता तुमची टेस्ट असेल. सो 20 आय विल सेंड यू द लिंक ऑन द ग्रुप. यू हॅव टू जस्ट जॉईन, गिव युअर रोल नंबर, नेम, those details you have to fill, and you have to take the questions, answers for 20 questions, 20 marks. Okay, and submit it. That's all you are supposed to do. So Google Forms ते तुमची टेस्ट होणार आहे. आणि सॅटर्डे यू विल फॉर tomorrow's test the syllabus will be only unit 1, and for Saturday's test your syllabus will be 2 and 3 unit. Today I'll be giving you the notes of second and third unit. Second unit I've already given, so third unit I'll be giving. So let us start with post-translational modifications. After translation, the protein product which is formed, it will undergo some modifications. And in the modified form, only that protein will be active. So that is why post-translational modification is nothing but covalent and enzymatic modification of proteins after protein synthesis. So we very well know that proteins are synthesized by the process of transcription and translation, and whatever protein product, the mature or active protein product is formed, it will undergo post-translational modification. This post-translation modification, it is very important component in cell signaling, because the proteins have to reach the sites where they show their action. So that is why in the modified form only the proteins can show their particular action. It is not necessary that in which form they are synthesized they will be always active. Like insulin's example आपण घेऊ शकतो. When insulin is synthesized, it is synthesized as a polypeptide chain. Insulin is a protein. It's a long chain polypeptide. But it has two chains. It has to be cut down into two chains. So that is why it is known as pre-proinsulin. It is cut down to form proinsulin by proteolytic activity, and again it is cut down to form the alpha and beta chains of the active insulin molecule. So that is pro-hormone conversion to hormones, pre-proinsulin, proinsulin, and insulin. So these three steps complete the process of post-translational modification in case of insulin. Similarly, in other proteins also various types of modifications take place at the carboxy terminal or the amino terminal of the protein. So this will modify the functional groups of the protein. Sometimes it's not necessary that only 5' and 3' end will be modified, but sometimes even the amino acids may be modified. Some phosphate group may be attached, some acetyl group may be attached, lipids, long chain lipid molecules may be incorporated. So different types of mechanisms are involved in the post-translational modification. So phosphorylation is the most common type of mechanism which regulate the activity of various types of enzymes, and it's the main type of post-translational modification, a very common type of post-translation modification. Then in eukaryotic and prokaryotic proteins, even glycosylation takes place. So glycosylation will be the addition of some sugar moiety or carbohydrate group to the protein. This will improve the stability and it will play a regulatory role. Then lipidation is one molecule in which lipids will be attached to the proteins, to the target proteins, and then the protein can attach to the cell membrane. So these are the various mechanisms we are going to see in the post-translational modification. First one is phosphorylation. So protein phosphorylation is the very common type of post-translational modification. In this case, a phosphate group is covalently attached to the amino acid. So phosphorylation takes place with the help of specific enzymes. They are known as kinases. And the reverse phosphorylation reaction is known as dephosphorylation, that is removal of phosphate group. And it is carried out by the enzyme known as phosphatases. So always you have to remember attachment of phosphate group or phosphorylation takes place with the help of enzymes called kinases, and dephosphorylation or removal of phosphates is done by the enzymes phosphatases. So phosphorylation of a protein, it is reversible event. It depends on the activity of kinases and phosphorylases. So it's a reversible type of process. Phosphorylation provides a quick, flexible, and transient signals that cells use to transmit the information, and they can respond to the stimuli. It is a common form of protein modification. It is used in signal transduction pathways. We are going to see those signal transduction pathways in the next unit. So where these type of phosphorylations are important for controlling various type of cellular mechanisms or processes involved in the cell cycle, like cell cycle progression, motility, metabolism, cell growth and differentiation, apoptosis, that is death of the cells after they age out, cellular communication, etc. Now, this reversible protein phosphorylation, it is found in case of serine, threonine, and tyrosine residues. They may be phosphorylated. And it is the most well-studied post-translational modification, that is phosphorylation of serine, threonine, and tyrosine. It helps in various types of mechanisms. Next one is glycosylation. Glycosylation is again one important protein, a post-translational modification, and it has various effects on the folding of proteins, the conformation of proteins, the stability of protein, the activity of protein. So ह्या सगळ्या केसेसमध्ये जेव्हा कार्बोहायड्रेट ग्रुप्स अटॅच होतात तर फोल्डिंग ऑफ प्रोटीनमध्ये त्याची मदत होते, त्याचा शेप क्रिएट करण्यासाठी किंवा कन्फर्मेशन क्रिएट करण्यासाठी त्याची मदत होते. काही प्रोटीन स्टेबल नसतील पण आफ्टर ग्लायकोसिलेशन दे विल बिकम स्टेबल. Some proteins may not show their activity, normal activity, but after glycosylations, their activity will be expressed. So, this is again an important effect observed after glycosylation, even distribution. So, where these proteins are present, that will again be decided by the carbohydrate moiety which is attached. So it is nothing but the addition of sugar moiety in various forms. It may be simple monosaccharides or modified types of branched polysaccharides to the protein. Carbohydrates in the form of asparagine linked or serine threonine linked oligosaccharides are the major structural components of many cell surface and secreted proteins. बऱ्याच ठिकाणी प्रोटीन्स हे सिक्रीट होत असतात, तर त्यावेळी कार्बोहायड्रेट्स कुठल्या फॉर्ममध्ये त्याच्यात अटॅच असतात तर दे आर अटॅच टू व्हेरियस अमायनो ॲसिड्स लाईक आर्जिनिन लिंक्ड oligosaccharide, serine linked oligosaccharide, or threonine linked oligosaccharide. So in this form they may be attached. Now this process of glycosylation can take place at various ends, like N-glycosylation at the amino end, O-glycosylation at the OH group. H will be removed and glycosyl group will be attached to it. Then glycation, here COH group वरती त्याला अटॅच होऊ शकतो. नंतर C glycosylation at the C terminal, glycosyl group may be attached, or phosphoglycosylation along with the sugar moiety, the phosphate group may be attached to the ore. So these are the various types of proteins with amino terminal, hydroxy terminal, COH group, or uh, glycosylation, carboxy terminal, sorry, then phosphate group is attached. So various types of glycosylations may be involved. Next type is ubiquitination. So ubiquitination किंवा ubiquitination पण म्हणतात त्याला. It's one and the same. Ubiquitination or ubiquitination. So it is again one enzymatic post-translation modification in which the ubiquitin protein is attached to the protein. Now, this process is commonly, in this case, the last amino acids of the ubiquitin, that is glycine, uh, it binds to lysine residue on the substrate. जोही सबस्ट्रेट असेल त्याच्या लायसिन रेसिड्यूला कोण अटॅच होतं युबिक्विटिनचा लास्ट अमायनो ऍसिड दॅट इज ग्लायसिन 76. What is ubiquitin? Ubiquitin is nothing but 8 kilodalton polypeptide which consists of totally 76 amino acids. And in these 76 amino acids, the last amino acid is glycine. Okay, and this glycine attaches to the lysine amino acid on the substrate on which this particular protein will attach. So where normally this ubiquitin binds, it binds to the amino terminal of the lysines in the target substrate or protein via C-terminal glycine of ubiquitin. म्हणजे सी टर्मिनल युबिक्विटिन पॉलिपेप्टाइडच्या सी टर्मिनलला ग्लायसिन असेल आणि जोही टार्गेट प्रोटीन असेल त्याच्या एन टर्मिनलला लायसिन असेल आणि हे दोघं बाइंड होऊन युबिक्विटिनेशन इव्हेंट होत असतं. So after the initial monoubiquitination, mono means only one ubiquitin molecule is attached. The formation of ubiquitin polymer may occur and several moieties of ubiquitin may attach to the protein, and that will result in polyubiquitinated protein. So these are recognized by 26S proteasome that catalyzes the degradation of युबिक्विटिनेटेड प्रोटीन. मग असा जो पॉलीयुबिक्विटिनेटेड प्रोटीन आहे तो प्रोटियाझोमने ब्रेकडाऊन होऊ शकतो आणि अगेन दॅट युबिक्विटिन कॅन बी रिसायकल्ड. आता युबिक्विटिन का अटॅच व्हायला पाहिजे तर त्या फॉर्ममध्ये तो पर्टिक्युलर साईट ऑफ ॲक्शनवरती ॲक्ट होऊ शकतो. म्हणजे ubiquitin will attach to the protein. In this form it will be taken to the target region. नंतर तो युबिक्विटिन तिथून रिलीज एक किंवा अनेक युबिक्विटिन मॉलिक्युल्स तिथे अटॅच होतात आणि दे कॅरी द प्रोटीन टू द पर्टिक्युलर साईट ऑफ ॲक्शन अँड देन दे कॅन किंवा पर्टिक्युलर ऑर्गनवरती किंवा कुठे सेल मेम्ब्रेनला जिथेही त्याची ॲक्शन असेल तिथे तो नेईल आणि नंतर दिस युबिक्विटिन विल बी रिलीज्ड अँड इट कॅन बी अगेन recycled to bind to another protein. That is known as ubiquitination. ह्याला ही सुद्धा आपण डिटेलमध्ये पाथवे स्टडी करणार आहे. Next is S-nitrosylation. So nitric oxide is produced by 3 isoforms of nitric synthetase. Nitric oxide synthetase. NO. It is produced from the 3 isoforms of nitric oxide synthase, and it is a chemical messenger which reacts with the free cysteine residues to form S-nitrothiols. So S-nitrosylation, again it is addition of NO, SNO, that is S-nitrothiols, to the particular protein. S-nitrosylation, it is again a critical post-translational modification. It is used by the cells to stabilize the proteins, to regulate the gene expression, and to provide nitric oxide donors wherever there is necessary. It is also involved in activation and catabolism of S-nitrothiols. Okay. So again, it is a reversible reaction. S-nitrothiols have a very short life in the cytoplasm, and host enzymes like glutathione, GSH, thioredoxin, and denitrosylate proteins, they can reduce this S-nitrosylation. So त्याच्यामुळे S-nitrosylation is a reversible reaction. It will bind to the protein, and again it will release the protein and can again recycle to bind to other proteins. Normally, S-nitrothiols are stored in the membranes, vesicles, interstitial space between the two cells, then in the cell membranes, in small vesicles, in the lipophilic protein folds, and they protect the proteins from denitrosylation. For example, caspases, which mediate apoptosis. Apoptosis means the death of the cell. And it is done. म्हणजे जेव्हा सेलची लाईफ संपते तेव्हा it kills itself. That process is known as apoptosis. Now this particular apoptosis is mediated with the help of caspases. They are stored in the mitochondrial intermembrane space as S-nitrothiols. So caspases वरती हे एस नायट्रोसिलेशनची रिॲक्शन झालेली असते आणि ते मायटोकॉन्ड्रियाच्या आतल्या साईडला आतल्या मेम्ब्रेनला अटॅच असतात. इन रिस्पॉन्स टू एक्स्ट्रा ऑर इंट्रासेल्युलर cues, the caspases are released into the cytoplasm. So काहीही एक्स्ट्रासेल्युलर और इंट्रासेल्युलर सिग्नल्स मिळाले की हे कॅस्पासेस सायटोप्लाझम मध्ये रिलीज होतात अँड दे डिनायट्रोसिलेट मीन्स द नायट्रोसिल ग्रुप एस नायट्रोसिल किंवा एस nitrothiols group will be removed from the proteins, resulting in caspase activity and induction of apoptosis. म्हणजे जोपर्यंत सेल लिव्हिंग आहे त्याची ऍक्टिव्हिटी नॉर्मली चालू आहे तोपर्यंत हे कॅस्पासेस ऍक्ट होणार नाही. सो त्याच्यावरती त्याला बाइंड करून कोण ठेवेल, एस नायट्रोसिलेशनची रिॲक्शनने एस नायट्रोथायोल तिथे बाइंड होऊन राहील आणि अशा फॉर्ममध्ये ते मायटोकॉन्ड्रियाच्या इनर मेम्ब्रेनमध्ये अटॅच राहतील. पण जेव्हा सेलची लाईफ संपलेली असेल त्यावेळी हे कॅस्पासिस सायटोप्लाझममध्ये रिलीज होतात आणि डिनायट्रोसिलेशन होतो. नायट्रोसिल ग्रुप त्याच्यातनं रिमूव्ह होऊन जातो. परत दॅट डिनायट्रोसिल ग्रुप that will S-nitrosyl group will go to bind to other type of caspases and now this caspases and can act on the cells and carry out the process of apoptosis. So again it is a reversible type of reaction. It is not a random type of reaction and only specific amino acid residues can bind to S-nitrosyl groups or they can be S-nitrosylated. Proteins consist of multiple cysteines and due to the labile nature of S-nitrosyls or S-nitrothiols, it is very difficult to detect these type of cysteines to which the S-nitrosyl group is attached. आणि खूप सारे ॲसेसच्या मदतीने आपण त्याला डिटेक्ट करू शकतो. सो व्हॉट विल हॅपन, एस नायट्रोसिल ग्रुपला डिटेक्ट केलं तर आपल्याला काय कळेल, कॅस्पासेस कुठे आहेत ते कळेल. मग त्याच्यामुळे आपण एपोप्टासिसची रिॲक्शन मीडिएट करू शकतो किंवा स्टॉप करू शकतो, वॉट एव्हर इज द need. So that is known as a biotin switch assay, which was first designed by Jaffrey et al. And it is the method which is used to detect the S-nitrosyl or S-nitrothiol groups in the cell. Next is methylation. It's nothing but the transfer of one carbon methyl group to nitrogen or oxygen. It may be N-methylation or O-methylation, depending upon the one carbon methyl group transferred to the nitrogen or oxygen. If it is transferred to nitrogen, we call it N-methylation. If it is transferred to oxygen, we call it O-methylation. So this increases the hydrophobicity of the protein, the affinity of the protein to the water molecule. It will not get dissolved in the water molecule. It can neutralize the negative amino acids charge when bound to carboxylic acid. चुकून सगळे प्रोटीन्स जर कार्बोक्सिलिक ऍसिड्सला बाइंड होतील तर नेगेटिव्ह चार्ज वाढून जाईल आणि ऑलरेडी नेगेटिव्हली चार्ज अमायनो ऍसिड्स असतील लाईक एस्पार्टिक ऍसिड, ग्लुटामिक ऍसिड, हे नेगेटिव्हली चार्ज अमायनो ऍसिड्स आहेत आणि ते कार्बोक्सिलिक ऍसिडला बाइंड होतील तर negative charge will increase. So to neutralize this negative charge, methylation can play an important role. So in this way, they will increase the hydrophobicity of the protein. This methylation is mediated by methyl transferase enzymes, and in presence of, it will take place in presence of S-adenosyl methionine known as SAM, and it is a primary methyl group donor. म्हणजे एन्झाईम्स कोणते लागतील? मिथिल ट्रान्सफरेसेस, आणि सबस्ट्रेट कोणता लागेल? एस ॲडिनोसिल मिथायोनिन, विच विल डोनेट द मिथिल ग्रुप to the particular protein. Methylation takes place so often that SAM has been suggested to be the most used substrate in enzyme reaction after ATP. म्हणजे मिथिल ग्रुप डोनर म्हणून एस ॲडिनोसिल मिथायोनिन खूप खूप सिग्निफिकंट आहे. तुम्हाला रिस्ट्रिक्शन मॉडिफिकेशन तुम्हाला जर आठवत असेल, जेनेटिक्समध्ये तुम्ही शिकले असाल मागच्या वर्षी. So in this modification also, S-adenosyl methionine acts as a methyl group donor. It will methylate the particular base, that is adenine or guanine, आणि तशा मिथिलेटेड बेसिसमुळे आपण रिपेअर मेकॅनिझम्स करू शकतो, किंवा मिथिलेटेड जे स्ट्रँड्स ऑफ DNA आहेत ते ब्रेकडाऊन होणार नाही किंवा रिपेअर होणार नाही. दोज विल बी द ओरिजिनल स्ट्रँड्स ऑफ DNA. So it will protect the DNA from the action of endonuclease type of enzymes. That is the main role of SAM or methyl group donor. N-methylation is irreversible, while O-methylation is reversible. So methyl group जर N अमाइन टर्मिनलला अटॅच असेल तर मग, किंवा नायट्रोजनला अटॅच असेल तर इट बिकम्स irreversible. But if it is attached to oxygen, it is a reversible process. So it is mainly involved in regulation, histone methylation, demethylation. It will help in the availability of DNA for transcription. म्हणजे एखादा स्ट्रँड जर मिथिलेट झाला तर तो ट्रान्सक्रिप्शनमध्ये इन्व्हॉल्ड नसेल, दुसरा त्याच्यामध्ये इन्व्हॉल्ड असू शकतो, किंवा मिथिलेटेड स्ट्रँड कॅन बी made available for the process of transcription. It's again a very important post-transcriptional modification. Then N-acetylation. So transfer of acetyl group to nitrogen is N-acetylation. It occurs in eukaryotic proteins. It may be both reversible and irreversible. N-terminal acetylation requires cleavage of the N-terminal methionine by methionine aminopeptidase. आपल्याला माहिती आहे प्रोटीनच्या N टर्मिनलला फर्स्ट अमायनो ॲसिड अमायनो टर्मिनल असल्यामुळे फर्स्ट अमायनो ॲसिड मिथायोनिन असेल, आणि यूकॅरियोट आहे तर तो मिथायोनिन फॉर्ममध्येच असेल. तर ॲसिटिलेशनच्या वेळी N टर्मिनल मिथायोनिन जो असतो तो अमायनोपेप्टिडेज, मिथायोनिन अमायनोपेप्टिडेज मॅप एन्झाईमने रिमूव्ह होईल किंवा क्लीव्ह होईल, आणि तो अमायनो ॲसिड मग ॲसिटिल ग्रुपने रिप्लेस होईल, ॲसिटिल कोए ने रिप्लेस होईल. दिस particular reaction takes place with the help of enzyme N-acetyl transferase, the NAT enzymes. So what is the mechanism? Addition of acetyl group to nitrogen. So this nitrogen will be present on the amino terminal of the protein. At the amino terminal of the protein, the first amino acid is methionine. So first step will be removal of that methionine by methionine aminopeptidase. Once this methionine is removed, its place will be taken by acetyl coenzyme A, and this addition of acetyl CoA to the N terminal will be taking place in presence of enzyme N-acetyl transferase. So this is co-translational mechanism. It may take place simultaneously during the translational process. N-terminal is acetylated on the growing polypeptide chain even when it is still attached to the ribosomes. बऱ्याच वेळा मिथायोनिन जशी ती पॉलिपेप्टाईड चेन तयार होत असते, स्टील रायबोसोमच्या एक्झिट साईट मधून रायबोसोमला अटॅच असेल ती पॉलिपेप्टाईड चेन, तेव्हाच ही प्रोसेस होऊ शकते ॲसिटिलेशनची. सो पहिला अमायनो ॲसिड मिथायोनिन असेल तो रिमूव्ह होऊन तिथे ॲसिटिल कोए अटॅच होईल, आणि ती अटॅचमेंट कोणाच्या मदतीने होईल? ॲसिटिल ट्रान्सफरेजच्या मदतीने. 80 to 90% of eukaryotic proteins are acetylated, आणि त्याचं मेन कारण मात्र अजूनपर्यंत कोणाला माहिती नाहीये. Acetylation of the epsilon amino of lysine on histone end termini is the common method of regulating gene transcription. So lysine acetylation ची पण process होते आणि that process is involved in the regulation of gene transcription. Histone acetylation however is a reversible event. यावरच्या जरी irreversible असल्या तरी histone acetylation, histones are proteins attaching to nucleic acid. So these histones they may be acetylated to reduce chromosomal condensation. म्हणजे खूप सारे DNA एकाच ठिकाणी कंडेन्स व्हायला नको आणि ट्रान्सक्रिप्शनची प्रोसेस सुटसुटीत व्हायला पाहिजे, त्याच्यासाठी हिस्टोन ॲसिटिलेशन प्रोसेस होत असते. Here lysine residues will be acetylated and this will regulate the accumulation of transcription factors, and mainly the enzyme involved here is histone acetyl transferase or HAT activity is involved. So transcription factors with this histone acetyl transferase activity, they can act as transcription co-activators. Histone deacetylase enzymes, they can act as co-repressors, which can reverse the process of acetylation, and that is why it is a reversible process. So lysine मधून तो acetyl group remove होऊन जाईल and chromosomes परत condense होऊन जातील. क्रोमोझोम्स किंवा DNA कंडेन्स झाला तर ट्रान्सक्रिप्शनची प्रोसेस नीट होणार नाही, सुटसुटीत पाहिजे DNA, सो दॅट दी एक्झॅक्ट RNA पॉलिमरेज और प्रमोटर साइट एक्सपोज होईल, RNA पॉलिमरेज तिथे बाइंड होईल आणि ट्रान्सक्रिप्शनची प्रोसेस इनिशिएट होईल. All these sites should be exposed, but if the chromosome is condensed, this may not take place properly. So lysine residues will be first acetylated, and by this acetylation the condensation of the chromosomes or DNA will be reduced. The DNA will be separated out for the process of transcription. Once this transcription is over, again the reverse reaction will take place, HAT activity will be reduced, and again the condensation of the chromosomes will take place. Sirtuins, that is silent information regulators. These are a group of NAD dependent deacetylases that target the histones. म्हणजे ॲसिटिल ग्रुप अटॅच व्हायला ॲसिटिलेज लागतो, पण रिमूव्हलसाठी परत डीॲसिटिलेसिसची गरज आहे. And these sirtuins, silent information regulators, these act as deacetylases for target histones. And gene silencing ची जी प्रोसेस आहे ती कशामुळे होते? हायपो ॲसिटिलेटेड हिस्टोन्समुळे. आपण मागे जीन सायलेन्सिंग बघितलेलं आहे. When acetylation was first detected in histones, cytoplasmic proteins are also acetylated. So फक्तच हिस्टोन ॲसिटिलेशन नसतं तर सायटोप्लाझममध्ये असलेले प्रोटीन्स पण ॲसिटिलेट होऊ शकतात, and these proteins help in the regulation of transcription. Then crosstalk between acetylation and post-translational modification, that is phosphorylation, ubiquitination, methylation, they overall modify the biological function of the proteins. Next one is lipidation. Again here, lipidation is addition of lipid molecules to the target proteins, and these proteins will be located in the membranes of various organelles like endoplasmic reticulum, Golgi apparatus, mitochondria, endosomes, lysosomes, plasma membrane ह्यांना ते अटॅच असतात प्रोटीन्स आणि त्यांचं लिपिडेशन होऊ शकतं. So होत असतं. So there are 4 types of lipidation mechanisms. C-terminal glycosyl phosphatidyl inositol anchor, GPI anchor त्याला असं म्हणतात. So at the C-terminal the lipids will be attached. N-terminal myristoylation, then S-myristoylation and S-prenylation. So one by one we'll see what it is. Each type of modification gives the proteins distinct membrane affinities. Although all types of lipidation, lipid आहे म्हणजे hydrophobicyशी connected असेल. Lipids are not soluble in water. So if the lipids is attached to the protein, it will give the hydrophobic property to that particular protein. आणि त्याच्यामुळे it will also affect the affinity of that protein for the membrane. So first one is DPI anchors. What is DPI? Glycosyl phosphatidyl inositol. Glycosyl phosphatidyl inositol anchors. So these act on cell surface proteins which are present in the plasma membrane. They will impart the hydrophobic properties, and if they are present in the membrane of endoplasmic reticulum, it will provide the for hydrophobic property to that particular newly formed protein. GPI anchored proteins, that is, addition of this glycosyl phosphatidyl inositol to the protein is known as GPI anchored protein. Where they are present, they are found in cholesterol, sphingolipid rich lipid graphs, and they act as signal platforms for plasma membrane. This is reversible. GPI can be released from the protein by special type of phospholipase enzymes. Next one is N myristoylation. So again, it provides hydrophobic property to the target protein. The myristoyl group, it is nothing but a 14 carbon saturated fatty acid, C14. 14 कार्बनची सॅच्युरेटेड फॅटी ऍसिड असते, जी प्रोटीनला अटॅच होते. वन्स इट अटॅचेस टू द प्रोटीन, इट गिव्स हायड्रोफोबिक प्रॉपर्टी टू द प्रोटीन, and it also affects its affinity to the membrane. But it can anchor this protein or attach this protein to the cell membrane. So affinity of the protein to the cell membrane increases by myristoyl group attachment, that is 14 carbon attachment to the protein. So it can also change the conformation of the proteins, and it can provide some binding surface to the protein, a handle like surface to the protein, which will help in the attachment of the protein to the membrane. So for example, signal proteins किंवा SRC family kinases, they are N-myristoylated. Then S-palmitoylation. Palmitoyl group is nothing but 16 carbon fatty acid group, palmitoyl coenzyme A, and it will be attached to the thiolate side, the S side of the cysteine residue. Cysteine मध्ये काय असतं? दोन सिस्टेन एस एच ग्रुप असतो. तर थायलोसाइड थायोलेट साइड म्हणजे एस एच ग्रुप, थायोल एस, थायोल रिफर्स टू एस. द सल्फाहायड्रल जो ग्रुप असतो, ती साईड जी असते त्याला आपण थायोलेट साईड म्हणतो. आणि सिस्टेनमध्ये एस ग्रुप्स असतात. तर त्या एस ग्रुपला हे C16 पामिटॉइल ग्रुप अटॅच होत असतो with the help of palmitoyl acyl transferases, PATs. So again, this will provide hydrophobicity to the particular protein. हे सिस्टेन कुठं असेल? त्या प्रोटीनमध्ये असेल, आणि त्या प्रोटीनच्या सिस्टेन ग्रुपच्या एस साईडला हा C16 पामिटॉइल ग्रुप अटॅच होईल, आणि त्याच्यामुळे तो प्रोटीन परमनंटली सेल मेम्ब्रेनला अटॅच होऊ शकेल किंवा अँकर होऊ शकेल. This can even be reversed. You can remove that particular protein from the cell membrane by thioesterases, which break the link between the protein and the palmitoyl group. So S-palmitoylation is used as an on-off switch to regulate membrane localization. S-palmitoylation is used to strengthen again other types of lipidation, that is myristoylation, farnesylation, etc. So we'll see what is farnesylation now. S-palmitoylated proteins are they selectively concentrate at the lipid rafts. So wherever hydrophobicity is required, these lipid rafts reach that position, आणि तिथे हे S palmitoyl groups attach होऊ शकतात तिथल्या प्रोटीन्सवर. Then S-prenylation. Here the farnesyl, that is C15, or geranyl, that is C20 group of specific cysteine residues ला ते अटॅच होतात. सो अगेन प्रोटीनच्या cysteine amino acids वरती farnesyl, that is C15 fatty acids, or geranyl fatty acids, that is C20 fatty acids, they may attach from the C terminal via the enzyme farnesyl transferase or geranyl transferase. If C15 fatty acids are attached, the enzyme involved will be farnesyl transferase. If geranyl C20 fatty acids are involved, then the geranyl transferases will be involved. So, apart from S-palmitoylation, S-prenylation is very stable process, and the resulting modified protein will be very stable. 2% of the proteins which are produced in eukaryotes are prenylated. They include all the members of the RAS superfamily, and farnesylated किंवा geranylated असू शकतात. काही प्रोटीन्स फार्नेसिलेटेड असतात, म्हणजे त्याला C15 फॅटी ॲसिड्स अटॅच असतील. काही जिरानिलेटेड असतील, म्हणजे त्याला C20 फॅटी ॲसिड्स अटॅच असतील. काहींना दोन्ही अटॅच असू शकतात. इन दॅट केस इट इज नोन ॲज फार्निसिलेटेड और जिरानिलेटेड प्रोटीन. सो कॉम्बिनेशन सुद्धा शक्य आहे. These proteins have 4 amino acid motifs at the C terminal, which determine the type of prenylation. कोणत्या टाईपचं प्रेनिलेशन झालेलं आहे, फार्नेसिलेशन झालेलं आहे की जिरानिलेशन झालेलं आहे, हे ते डिसाईड करू शकतात. Prenylation occurs in the endoplasmic reticulation, and it takes place in a stepwise manner. In case of post-translational modification, it is followed by proteolytic cleavage by RCE1 and methylation by isoprenyl cysteine methyl transferase. त्याला रिव्हर्स पण करता येऊ शकतं बाय दीज टाईप ऑफ एन्झाईम्स. प्रोटिओलायटिक क्लिवेज म्हणजे एक तर प्रोटीन काढून टाकायचा त्या C15 C20 फॅटी ॲसिड्समधून, किंवा मिथिल ट्रान्सफरेसच्या मदतीने हे फॅटी ॲसिड्स सेपरेट करू शकतो आपण. You can see this diagram, it will give you the idea of various types of post-translational modifications which take place. So, P जिथे जिथे आहे, that is phosphorylation. नंतर ME जिथे आहे तिथे मिथिलेशन. ओके, वेगवेगळे टाईपचे आहेत. UB असेल तिथे युबिक्विटिनेशन किंवा युबिक्विटिनिलेशन. आणि ॲसिटिल ग्रुप सुद्धा अटॅच्ड आहे. सो दॅट इज ॲसिटिलेशन. हे 4 टाईप्सचे रिॲक्शन्स या एक डायग्राममध्ये, हा एक प्रोटीन आहे आणि त्याच्यामध्ये ॲसिटिलेशन, मिथिलेशन, फॉस्फोरिलेशन आणि युबिक्विटिनेशन 8 टाईम झालेले आहे. सो दिस फिनिश your post-translational modification, very significant process in case of protein synthesis. याच्याशिवाय आपण म्हणू शकत नाही की प्रोटीन सिंथेसिसची प्रोसेस कंप्लीट झालेली आहे. सो हिअर वी फिनिश विथ द 3rd युनिट. अजून 2 युनिट बाकी आहेत तुमचे. सो टुमॉरो विल स्टार्ट विथ द नेक्स्ट युनिट, अँड प्लस रेग्युलर तुमचं लेक्चर होईल, आणि त्याच्यानंतर मग तुम्ही हे करू शकतात, टेस्ट देऊ शकतात. म्हणून मी 2:30ची वेळ ठेवलेली आहे. 2:30 ते 3 मध्ये तुम्ही ते कंप्लीट करायचं. 30 मिनिट्स आर सफिशियंट फॉर 20 क्वेश्चन्स. सो विदिन 30 मिनिट्स 2:30ला टेस्ट शार्प सुरू करायची, 3ला पूर्ण करायची. 5 मिनिटं आधी मी तुम्हाला लिंक देईल, फटाफट तुम्ही भरायचं, सॉल्व करायचं, आणि 3 वाजता तुमची सबमिट झालेली पाहिजे. 3 नंतर जे सबमिट करतील त्यांना डिस्क्वालिफाय, मी ते धरणार नाहीत मग. ठीक आहे. सो फर्स्ट टेस्ट विल बी गिविंग ऑन द फर्स्ट युनिट टुमॉरो. क्लिअर? तुम्हाला क्वेश्चन्स दिले का मी? डिड आय गिव्ह यू द क्वेश्चन्स ऑफ युनिट 1 MCQs? येस और नो, पटापट उत्तर देत जा प्लीज. बी फास्ट. आर यू लिसनिंग और नॉट? यू शुड बी व्हेरी फास्ट इन आन्सरिंग. Did I give you the questions of unit 1st? No. So you just revise the notes and answer the questions tomorrow. Okay? Unit 1चे notes तर दिलेले आहेत, ते रिवाइज करा. त्याच्यातनंच काही क्वेश्चन्स असतील सो सॉल्व दोज क्वेश्चन्स. 20 मार्क्सचे 20 क्वेश्चन्स असतील आणि 30 मिनिट्स तुम्हाला वेळ दिला जाईल. एनी डिफिकल्टी, एनी प्रॉब्लेम? फटाफट आन्सर करायचे. एनी प्रॉब्लेम, एनी डिफिकल्टी? So we'll stop the lecture here, and tomorrow I'll start with the 4th unit plus test, plus Shruti is going to give her exam at 12 o'clock. 12 वाजता श्रुती द्यायची exam तुला. Let us stop here. We'll continue it.